Bismillah ar-Rahman ar-Rahim. The title of our lecture is the Major Histocompatibility Complex, and the objective of this lecture is to know the definition of MHC, which is the abbreviation of Major Histocompatibility Complex, and the structure and the function and the tissue typing. And the major histocompatibility complex, which is present in each mammalian species, and it is a cluster of genes, and their products play a major role in cellular recognition and in the determination the transplanted tissue is accepted or rejected. The product of these genes are expressed as antigens on the cell surface of the cells, and also called the human-leukocyte antigen, and they located on the short arm of the chromosome 6. So this is the chromosome 6. This is the short arm of the chromosome, and here the cluster of genes, and they divided into 3 classes: class 1, class 2, and class 3. This is the long arm of the chromosome 6, and this is the short arm of the chromosome number 6, and here the centromere, and here the cluster of genes on the short arm of the chromosome number 6, which is called the MHC, Major Histocompatibility Complex. And this is if we amplify this, it's divided into 3 classes: class 1, which is A, B, and C, and class 2, which is the RDP, DQ, and the class 3, which is C2, C4, and protein factor. And the class 1 is presence on all nucleated cells and platelets, while class 2 is presence on B cells, macrophage, and dendritic cells. The major histocompatibility complex, containing more than 220 genes, encoded the hyperpolymorphic 3 classes: class 1, class 2, class 3, and many of which contribute to immune defense against infection and influence the outcome of the organ transplant. First, we will deal with the class 1. It's of 2 types: classical, the class 1, which is include A, B, C, and the non-classical class 1, which is include E, F, G, H. And here the structure of the MHC class 1, which is consists from the single polypeptide chain, which is alpha 1, alpha 2, and alpha 3, and bind non-covalently with the beta microglobulin. And this is the extracellular part of this class, and this is the transmembrane, and here the intracytoplasmic part. And the alpha 1 and alpha 2 form a basket-like structure to hold the epitope of the antigen. As we mentioned, the antigen is consisted from multiple epitopes, and here will bind to the single epitope of the antigen. And this MHC class 1 distributed on all nucleated cells and the platelets. Cellular distribution of the MHC molecules, they are expressed on the most somatic cells, T lymphocyte, which is highest level of the expression, and all nucleated cells and platelets. Now we have the class 2 structure of the MHC. It consists from the 2 polypeptide chain, alpha chain and beta chain, and the alpha chain, which is so long that it is folded to form a domain, which is alpha 1 and alpha 2 and beta 1 and beta 2. And this is the extracellular structure, and this is the transmembrane and the intracytoplasmic part. The class 2, the alpha 1 and beta 1 domain form a membrane distal groove that holds the epitope of the antigen. And the distribution of the MHC class 2 is distributed on the B cell, macrophage, dendritic cells, and the all antigen presenting cells. The domain alpha 1 and alpha 2 in class 1 and alpha 1 and beta 1 in class 2 interact to form a plate of 8 anti-parallel beta-pleated strands by 2 long alpha helical regions. And this is the structure that holds the epitope of the antigen. This forms a deep groove with alpha helix at sides and beta-pleated sheet as the bottom. This is the structure of the class 1. This is the beta-pleated sheet anti-parallel. This is the alpha 1 and this is the alpha 2. While in the class 2, the alpha 1 in the yellow color and the beta 1 in the green color. And here the groove that binds the epitope of the antigen, and here the groove or the basket that binds the antigen. And the variable region responsible for the HLA polymorphism lies along the alpha helix in the class 1, while in the class 2 only in the beta 1. This is the red one. When there is single amino acid change, so there is change in the structure of the basket that bind to the epitope of the antigen. The function of the MHC presentation of the antigen, which is important in the development of humoral and cellular immune response. As you know, the humoral with the class 2 and the cellular with the MHC class 1. And here the class 1, which is the antigen presenting cell, which is presented to T cell. This is the MHC class 1 and this is the MHC class 2. MHC have been implicated in the susceptibility also to disease, especially autoimmune disease, and MHC is important in the inflammation, infection, and autoimmune disease. It's also important in the transplantation, and it's associated with pharmacogenetics like hypersensitivity to drugs like abacavir, which is induced liver injury in the HLA 5701 bearing individuals, and it's also important in the human population genetic study. The MHC are highly polymorphic, so each person did not the MHC of each person is not similar to another person. So it is highly polymorphic, which is that we could say many alternative forms of genes or alleles exist at each locus. Locus is a position of gene on the chromosome, while the allele is the alternative forms of a gene at a single locus. Here the alleles, the class 1 had 3 locus A, B, C, and this is the A, and the due to high number of the alleles, they give it numbers 1, number 2, number 3, number 9, etc. And this is the A is locus, B is locus, C is locus, and this is the alleles. They give it the numbers to differentiate one person from the another, and this is C locus and this is the alleles. The same thing with the class 2, and this is DR, DQ, and DP. Also, this is the locus and here the alleles. So different HLA alleles, class 1 had more than 451 alleles, B more than 700, C more than 2, and same with the HLA class 2, DR, DQ, DP, so there is many alleles. The ontology of the HLA alleles has been continuously developed since 1968, and the immune polymorphism database is a set of specialist databases related to the study of polymorphic genes in the immune system. It's developed to provide a centralized system for the study of polymorphism in genes of the immune system. Due to high polymorphic range of this MHC, So they develop what is called database to these alleles. Inheritance of the MHC molecules. Each individual inherits a complex set of alleles known as haplotype, encoded by closely linked alleles, one from the mother and the other from the father. And these alleles are codominantly expressed, and there is no recessive. They are codominantly expressed. And here the leukocyte, and here the MHC, which is encoded on the cell surface of the leukocyte, and here the set from the mother, and here the set from the father. And for example, here the father mate with the mother, and they have children, and this is the HLA of the father, and this is the HLA of the mother, and the HLA of the 25% of the children, A1, 51, and 2, 7. And this is the haplotype. We read it horizontally. AB, AB. 1, 51, 2, 7. This is from the mother, this haplotype from the mother, and this haplotype from the father. And this constitutes 25% of the offspring. And the another 25% of their children have the another haplotype, 28, 27 from the mother, and 33, 18 from the father. And the another 25% of their children, their HLA typing is 1, 27, and 2, 18. And the rest, 25% of their children, 28, 51, and 33, 7. This chiasmatically, the inheritance is called crossover genetic recombination. There is a chiasmatically inheritance, which is rare. So this follows the Mendelian laws of genetic, which is 25% of siblings share 2 haplotypes, 50% of siblings share 1 haplotype, and 25% of siblings share no haplotype. This is Mendelian law. Thus, two random individuals are unlikely to have identical set of HLA molecules. So each one of us, HLA typing of him is not similar to another person. So reject organ transplantation and differ in their susceptibility to disease. Now we have a new subject, HLA nomenclature. How can nominate the HLA typing? According to method, we did the test serological nomenclature, which is the HLA, and this is the locus, and this is the allele. While in the molecular nomenclature using DNA typing, this is the locus, and we put a star. When we see a star, so once we see a star, so we can understand that this HLA typing done by molecular method. And here the allele group, and here the allele number, and this is the allele group, which is similar to serological method. And here the allele number, and here the silent polymorphism in exon, and also give it a number. And this is gene coding for beta chain. This is for beta chain. When there is A, so for the alpha chain. Genetic polymorphism. Why there is large number of HLA alleles, and this is due to genetic recombination, crossover, as here. One segment translocated to the another segment. So this leads to production of new allele. Intra-allelic conversion, and here the rotation of the chain of the DNA, and gene conversion. The gene, there is a conversion in it. And last one, the mutation. All these factors lead to large number of HLA alleles. Now the new subject is tissue typing. What's tissue typing? Is a laboratory test used to determine the type or the number of alleles to each person in each locus. HLA typing done by following methods: serological method and molecular method. And molecular method of two types, low resolution and high resolution. Serological method done first by scientist Terasaki and McClellan in 1964, while molecular method genotype level uh, of many types, SSP, which is sequence-specific primer, and another method, RASCA, which is reference transconformational analysis, and the newest one, which is NGS, next generation sequencing, and DNA sequencing. Sometimes you use the letter W, which is mean workshop, and this is rarely used because of the technique, molecular technique, and this is mean that the allele is not well recognized and still under study. So thanks for NGS that leads to change in analysis HLA locus due to its highly accuracy. Uses of HLA typing. First, paternity testing in forensic medicine to prove the identity of illegal baby. And disease association, most common disease, Bahjat disease with HLA-B5 and ankylosing spondylitis with HLA-B27. Organ transplantation, which is very, very important, kidney, liver, heart, lung, etc. Control immune response by MHC restriction. Class 1 present antigen to cytotoxic T cell, while the class 2 present antigen to T helper T cell. Anthropology, study race and nation, because Caucasian is different from African, different from Americans. So the race, each race has its own HLA typing. Causes of MHC. And why there is what is the control of the HLA alleles production? First, transcription factor, class 2 transcription factor. This is class 2 transcription A factor. Regulatory factor X. Defect in these two factors leads to bare lymphocyte syndrome. Second, cytokine, which is alpha interferon, beta, gamma, tumor necrosis factor, interleukin 4. Gamma interferon increase the class 2 expression. Drugs, corticosteroid and prostaglandins decrease expression of class 2 molecules. So use cortisone in high dose lead to decrease in immunity, and this is due to decrease expression of class 2 molecules. Causes of MHC and disease susceptibility. There are a number of different diseases associated with a particular HLA alleles. As we said previously, HLA-B27 and ankylosing spondylitis, and B5 with Behçet disease. And why these associations occur between the HLA alleles and these diseases? First, molecular mimicry between HLA antigen and given pathogen. So the immune system, instead of attack the pathogen, will attack the HLA antigen. Second, immunologic cross-reaction in infected individuals. Third, contribution of linkage disequilibrium, linkage between HLA ABC with the RDQ, close linkage of tumor necrosis gene with HLA-B locus, tumor necrosis factor gene, which is present in the class 3, which is close to the class 2. MHC, another reason, MHC genes encode molecules that serve as receptors for the pathogen. There are the second part, which is the non-classical. We said previously all the classical part of the class 1 Now we have the non-classical class 1 type, which is include 4 locus, which is recently discovered, E, F, G, H. Their functions were unknown except HLA-G, and these are also polymorphic, and with time they increase in its numbers, like the classical type. And the HLA-G expressed on the cytotrophoblast at the fetal-maternal interface that protect fetus from being recognized as foreign and leads to abortion. And the another function, HLA-G exerts tolerogenic function involved in the transplant acceptance. And lastly, HLA-G was important in tumor and viral immune escape. And this is the most function of the HLA-G. And thank you.